A continent, millions of people. Village after village, from the high Andes to the lowland Amazon, from the deserts of the American Southwest to the frozen edge of the Arctic. You run the numbers, you run them again. In population after population, the result is the same. One blood type, nearly every single person. No exceptions that fit any pattern. No gradient, no transition zone, no neighboring community with a different profile. A hard wall. A biological uniformity that shouldn't exist at that scale, and that no one for a very long time could explain. The type is not defined by what it has, it is defined by what it lacks. It carries no A antigen, no B antigen, just an absence, a silent deletion baked into the DNA that science spent over a century treating as the neutral option, the unremarkable default, the baseline from which everything else departed. That assumption has been quietly unraveling for decades, and what the data has revealed, piece by piece, is that this absence is anything but neutral. Blood type O carries a record, a story, a set of evolutionary decisions so ancient and so consequential that they predate not just written language, not just civilization, but, as it turns out, our species itself. In the early 20th century, a Viennese physician named Karl Landsteiner made one of the most consequential discoveries in the history of medicine. In 1901, he identified the ABO blood group system, the molecular reason why some blood transfusions save lives and others kill. He received the Nobel Prize in Physiology or Medicine in 1930, and the framework he established became the foundation of modern transfusion medicine. The science, as it was taught for generations, went like this. Blood type is determined by a single gene, the ABO glycosyltransferase gene, located on chromosome 9, which acts like a molecular instruction manual. Think of it as a factory specification. Type A tells the body to build one kind of surface marker on red blood cells. Type B tells it to build a different one. Type AB builds both. And type O? Type O carries a mutation, technically a deletion, a single missing letter in the genetic code that stops the factory from building anything at all. No markers, no antigens, just bare cell surfaces. This made type O the universal donor. Because type O red blood cells carry no A or B flags, they can be transfused into patients of any blood type without triggering an immune rejection. For most of the 20th century, that was the headline. Type O, generous, simple, the base state from which the others evolved. The global picture seemed to support this framing. Type O is the most common blood type on Earth, carried by roughly 45% of the global population. Type A dominates in Northern and Central Europe, reaching 45 to 50% in Scandinavia. Type B is most prevalent in Central and South Asia. In parts of India and Pakistan, it exceeds 30 to 35% of the population. Type O, meanwhile, is common across sub-Saharan Africa and overwhelmingly dominant in the Americas. The map looked like a reasonable distribution of a neutral default type, spread wide by ancient migrations. But the textbooks never quite resolved the question hiding at the edge of that map. If type O is truly neutral, if it carries no functional advantage, if it is simply the version of the gene that does nothing, why is it found at near total exclusion in specific populations? Why would a neutral default become the only blood type in millions of people across 2 entire continents? The absence of an explanation was itself an anomaly, and science, eventually, started paying attention. Here is where it gets complicated. When researchers began systematically mapping ABO blood type frequencies across indigenous populations of the Americas, the numbers they found didn't fit any standard model of genetic variation. In certain Quechua communities in the high Andes of Peru, type O frequency reached 99%. Among the Yanomami of the Amazon basin, it was approximately 92%. Among the Navajo of the American Southwest, similarly extreme concentrations. Group A was virtually non-existent in Central and South American indigenous populations. Not rare, not uncommon, essentially absent, as if it had been erased. Not a gradual gradient, a wall. This alone was puzzling. But what made it harder to dismiss as a statistical quirk was a parallel anomaly emerging from a completely different region of the world. In 2007, a team at the University of Edinburgh published a study in the Proceedings of the National Academy of Sciences. Lead researcher A.M. Rowe and colleagues enrolled 567 Malian children, some with severe malaria, some with uncomplicated malaria, some healthy controls, and measured their blood types. The result was striking. Type O was present in only 21% of severe malaria cases, compared to 44 to 45% of healthy controls. Blood group O was associated with a 66% reduction in the odds of developing severe malaria. Not a marginal effect, a near halving of the risk. The mechanism remained unclear until 2015, when Professor Mats Wahlgren at the Karolinska Institutet in Sweden led a team whose findings were published in Nature Medicine. What they found was conceptually elegant. The malaria parasite, Plasmodium falciparum, uses a protein, a rifin variant, to make infected red blood cells stick to uninfected ones, forming clusters called rosettes. Think of rosetting like a blood cell pileup on a highway. Clumped cells block the tiny vessels of the brain, causing the cerebral complications that kill. Type O cells resist this rifin binding. The clusters don't form. The highway stays clear. Our study ties together previous findings, Professor Wahlgren said at the time. We can explain the mechanism behind the protection that blood group O provides against severe malaria, which can in turn explain why the blood type is so common in the areas where malaria is common. So, in Nigeria, over half the population carries type O. In malaria-endemic sub-Saharan Africa broadly, type O dominates. The disease map and the blood type map overlap with a precision that is difficult to attribute to coincidence. A deletion mutation, the absence of an antigen, had become, in regions ravaged by malaria, an invisible shield. If type O is truly just the version of the gene that does nothing, the data requires an explanation for why doing nothing kept so many people alive. The first theory says the answer is malaria. Full stop. Malaria is not a modern problem. It is one of the oldest and most relentless selective pressures in human evolutionary history. The WHO estimates it still infects 200 million people annually in the modern world. In prehistoric and ancient populations, particularly in equatorial Africa and South Asia, it was likely among the leading causes of death across hundreds of thousands of years. Under those conditions, even a modest survival advantage compounds quickly across generations. Type O carriers in malaria-endemic zones survived at slightly higher rates, reproduced slightly more often, and over millennia, their blood type spread through the population, not by design, not by migration, but by the brutal arithmetic of who lived long enough to have children. The geographic correlation is the strongest evidence for this theory. Place a map of historical malaria endemicity next to a map of global type O frequency. The overlap is not perfect, but it is substantial. In malaria-free Northern Europe, type A dominates. In malaria-endemic West Africa, type O dominates. The pattern is consistent enough that many researchers treat it as essentially settled. But here is the limitation. This explanation works for Africa. It works for parts of South Asia and Southeast Asia, where malaria has long been endemic. It does not work for the Americas. The Yanomami of the Amazon basin carry type O at 92%. The Quechua of the high Peruvian Andes, at elevations above 3,500 meters where temperatures kill mosquitoes, carry it at 99%. These populations had no significant burden of Plasmodium falciparum malaria before European contact. The disease arrived with the colonizers. It was not a shaping force in their pre-contact evolutionary history. Yet they show the same extreme type O concentrations as West African populations that have lived with malaria for tens of thousands of years. The malaria hypothesis explains part of the map. It does not explain the wall in the Americas. Something else was operating there. The second theory is more complicated, and it requires going back much further in time. The Beringian standstill hypothesis holds that the ancestral population of all Native Americans spent a prolonged period, possibly thousands of years, in the region of Beringia, the land bridge between Siberia and Alaska, before crossing into the Americas roughly 15,000 to 20,000 years ago. This founding group was small, extremely small, and small populations carry the DNA equivalent of a sampling error. If the founders happened to carry predominantly type O alleles, then when they expanded across 2 continents and eventually numbered in the tens of millions, type O would spread with them, not because it was advantageous, but because it was all they had. This process is called genetic drift, the random propagation of whatever alleles a bottleneck population happens to carry. Think of genetic drift like a small bag of marbles. If you pull 10 marbles from a barrel of mixed colors, and all 10 happen to be red, the next bag you fill from those 10 will be mostly red, not because red marbles are better, but because that's what you pulled. The evidence for this bottleneck is encoded in a specific genetic marker. Indigenous populations of the Americas carry a distinctive haplotype, O1V G542A, that is found in Native American populations at frequencies ranging from 4 to 60%, but is virtually absent in their Asian source populations. This suggests the type O fixation in the Americas was not simply inherited from Asian ancestors. It carries a genetic signature that mutated or was selected during the Beringian standstill itself. Something happened in that frozen corridor that shaped the blood type profile of an entire hemisphere. And then came a finding that pushed the timeline further back than anyone expected. In 2009, researchers at the Max Planck Institute for Evolutionary Anthropology, led by Carlos Lalueza Fox, analyzed DNA from 2 Neanderthal individuals buried at the El Cidrón site in northern Spain. Both individuals carried the O01 allele, the same type O deletion found in modern humans. The O mutation, in other words, predates the divergence of Homo sapiens from Neanderthals. It is at minimum 500,000 years old. Some estimates place the coalescence of the O01 allele at over 1 million years. This blood type is not a modern adaptation. It is older than our species. But here's where the second theory too runs into its wall. Founder effect explains why type O would become fixed in the Americas after a small founding population crossed Beringia. It does not explain why O remains the most globally widespread blood type on Earth after tens of thousands of years of population mixing, expansion, and contact. A truly neutral variant, drifting by chance, should have eroded back toward random distribution as populations merged and diversified. Instead, it held. Something is maintaining it. Something is continuing to favor it, even after every bottleneck and every migration event that should have diluted it back into the statistical noise. This is the part the records don't explain. The third theory, and the one most researchers are hesitant to say out loud, is that type O is not a single story. It is a different story in every population that carries it. And the force maintaining it is not one disease. It is all of them, all at once, in different configurations. Type O resists severe malaria. That is confirmed. But the same blood type that protects against malaria makes its carriers more vulnerable to severe cholera. Vibrio cholera disproportionately damages type O individuals, a finding that is well established in the literature on infectious disease and blood group antigen biology. The protection and the vulnerability are inseparable. Type O did not evolve to beat malaria. It evolved into a compromised position in an ongoing multi-front evolutionary negotiation between the human immune system and every pathogen it has ever faced. More recently, early data from the COVID-19 pandemic added another dimension. A study using records from the University of Chicago Medicine covering 2020 to 2021 found that blood type B was associated with significantly elevated susceptibility to SARS-CoV-2 compared to blood type O, suggesting that pathogen-specific interactions with ABO antigens continue to shape disease outcomes in real time. The arms race did not end in prehistory. It is still running. Researchers are careful to frame this as probability, not certainty. But the implication, if the data holds up at scale, is that the global distribution of ABO blood types reflects which pathogens historically dominated each region, not which blood type is universally better. Type O did not win. It survived a specific set of enemies while being vulnerable to others. In a world with different diseases, a different blood type might have dominated. The data doesn't confirm this fully. It only fails to rule it out. And what it leaves open is a possibility more unsettling than a simple adaptive story. That blood type frequencies in every human population are still shifting under pathogen pressure right now, in response to diseases that are still evolving. that the distribution we call normal is a snapshot of an ongoing process we are only beginning to measure. Imagine you are standing in the Peruvian Andes in the year 1490. The altitude is approximately 3,500 meters. The air is thin and cold, and it carries the smell of wood smoke and damp stone. The sun is dropping behind a ridge to the west. Somewhere below you, a river is audible but not visible. A child has been born in this village tonight. The parents do not have a word for blood type. No one in this village does. No one in any village does, anywhere on Earth. The concept will not exist for another 411 years. The parents hold this child and they know nothing of ABO glycosyltransferase genes or chromosome 9 or deletion mutations. They know this child is alive. What they cannot know, what no one will be able to say until the 21st century, is that this child carries no A antigen, no B antigen, just the silent absence that has been passing through this family's bloodline since before their ancestors crossed a frozen land bridge. Every person in this village carries the same marker. It has been here unspoken for millennia. In 5 years, European ships will make landfall on the coast. In the decades that follow, disease will move across this continent faster than any army. And in communities that had somehow maintained non-O blood types, the small minority, researchers will later find evidence of higher mortality rates from certain pathogens. Not because those people were weaker, because they carried different antigens in their blood, and those antigens gave some diseases a slightly better grip. The child in this village does not know any of this, neither do the parents. A fire burns at the center of the village. Breath is visible in the cold air. Here is the twist. It requires setting aside the story you were just told. For over a century, blood type O has been described as the universal donor, the type that gives freely, that asks nothing, that can be given to anyone. That framing made type O sound generous, neutral, self-sacrificing. The medical utility is real, but the evolutionary story runs in precisely the opposite direction. Type O exists because type O took. It took survival advantage from malaria. It took dominance from a population bottleneck. It took centuries of reproductive edge in disease environments that punished other blood types. The type defined by its absences outcompeted the types defined by what they carry. The deletion spread wider than the instructions. And now comes the deeper reframe. For most of scientific history, it was assumed that type O was the ancestral baseline, the original human blood type, the one from which A and B diverged, the neutral default, the starting point. Phylogenetic analysis has since established that this assumption was wrong. Type A is the most ancient human ABO allele. Multiple evolutionary studies have confirmed that type B diverged from type A approximately 3.5 million years ago, and that the O allele, the deletion, the absence, emerged later, as a mutation of the A allele. The O allele's most common variant, O01, is estimated to have arisen roughly 1.15 million years ago. Type O is not the original. It is a relatively recent edit, an absence that was introduced into the genome long after the presence was already there, and then, in specific environments, began to outperform its source material. The type that carries nothing built from what was already there, stripped the blueprint, kept the benefits, and spread. Not ancient, not primordial, edited. Pull back far enough and the picture becomes something larger. Your blood type is not a personality. It is not a diet prescription. It is not a destiny. It is a record, a compressed molecular archive of every epidemic, every population bottleneck, every migration corridor that your ancestors survived. The frequency of type O in Nigeria reflects the long history of malaria on the West African coast. The near total dominance of type O in the Andes reflects a founder population that crossed a land bridge and never looked back. The ongoing vulnerability of type O to cholera reflects a different battle that O did not win cleanly. Every distribution is a ledger. Every population carries in its blood type frequencies a partial accounting of what tried to kill its ancestors. The full accounting is still incomplete. The malaria pressure explains part of it. The Beringian bottleneck explains part of it. The cholera paradox raises questions that don't resolve neatly. And the Neanderthal data at El Cedrone reminds us that the mutation at the center of this story is older than modern humans, that we inherited this particular absence from a species that no longer exists, and have been living with its consequences ever since. A researcher who has spent years working on abiogenetics described it this way: there is a temptation to look at blood type frequencies and find a clean story, a single cause, a winning adaptation. But what the data actually looks like, she said, is a palimpsest, layer after layer of different pressures, different diseases, different migrations, written over each other until you can barely read the original. Scientists have finally revealed why O blood type carries a secret no one expected. But the deeper question, the one the data keeps opening rather than closing, is whether that secret has finished writing itself.